Hello students, welcome to EPG Pathshala. I am Monica Saini from Department of Anthropology, University of Delhi. Today I will talk about the module Identification through Hair under Forensic Anthropology paper. At the end of this module, you will be able to understand the morphological and structural characteristic of hairs such as modulation, diameter, form and cross section with their types and formation. You will be also able to understand the use of hair strand in identification at the scene of crime. This module will also tell you about the identification process to determine age, sex, blood groups, body area, and race. Human hair has a significant potential in forensic anthropology, and it has been extensively used in forensic investigations. Investigators believe that their presence can associate a suspect to a victim or a suspect or victim to a crime scene, primarily because hair can be transferred from the suspect to the victim and vice versa during physical contact. The types of hair recovered, the condition and number of hair found at the scene of crime all have great relevance as evidences in criminal investigation. The forensic anthropologists, they routinely compare the morphological characteristics of the hair sample in question to known hair sample to determine a transfer. Human hair has also been successively used to assess drug and substance abuse, as drugs of abuse cannot be often detected in body fluids. Using very sophisticated analytical techniques such as immunoassay, gas chromatography, the investigators are now extensively using human hairs to solve cases of poisoning, drug and substance abuse. Hair may be defined as slender filamentous outgrowth of the skin and primarily composed of keratin protein. Human hairs differs from one animal species to another in the basis of their length, color, shape, root appearance and morphological characteristics. Hair is composed of the protein which is known as keratin. It is also the primary component of the finger and toenails. Hair is produced from a structure called the hair follicle. Humans, they develop hair follicles during fetal development and no new hair follicles are produced after the birth. Hair color is mostly the result of pigments, which are chemical compounds that reflect certain wavelengths of visible light. Hair shape that may be round or oval, and the hair texture, which could be curly or straight, is heavily influenced by genes. The physical appearance of hair can be affected by nutritional status and intentional alteration. This intentional alteration, it could be heat curling, perms, straightening, or it could be anything else which alter the hair texture. The body area, head, arm, leg and back from which a hair originated can be determined by the sample's length, shape, size, color and other physical characteristics. In order to test hair evidence for nuclear DNA, the root must be present. The hair may also be tested using mitochondrial DNA whether or not the root is present. Hair is a biological polymer with over 90% of its dry weight being made up of keratin protein. The keratins are cystine containing proteins which form two large groups called intermediate filament proteins and intermediate filament associated proteins. These proteins are almost equally abundant in most hair. Of the several types of the bonds that stabilizes the keratin molecule, the unique linkage is formed by two cysteine residues contained in adjacent polypeptide chains, and it is this disulfide bond that is mainly responsible for keratin's resistance to destruction. The human hair follicle is a structure which is formed as a result of the epithelio-mesenchymal interactions. These interactions initiated around the third month of the fetal development. It is very complex, consisting of more than 20 different cell types distributed into 6 main areas. These areas are the connective tissue sheet, the dermal papilla, the outer root sheet, the inner root sheet, the shaft, and the sebaceous gland. This complex appendage behaves in a unique pattern in mammals, as after a hair production phase, it involves or it involutes in situ before entering a resting phase, after which it renews in a cyclical but stochastic fashion. The hair follicle thus is a fully autonomous skin appendage with its own hormonal control, its own autocrine and paracrine network, and its own cycle, appearing as an incredibly complex and stable structure that summarizes the main rules of the tissue homeostasis. Hair consists of a bulb or root and shaft. In hair, there are well-defined layers consist of cuticle, cortex, and medulla. As you can see in this diagram, the cuticle is the outer layer and it consists of thin, non-pigmented scales, whereas the cortex is the middle layer consisting of longitudinally arranged, elongated cells without nuclei. This cortex part is protein-rich structure around the medulla and it is mostly pigmented. Within these cell fibrils or on which there may be granules of pigments. The last layer is the medulla, which is also known as the central core. This is the inner layer composed of the keratinized remains of these cells, and sometimes this medulla layer it remain absent. The major constituents of the hair consist of keratin, melanin, and redheads. The keratin is a protein that makes up most of the hair shaft. The melanin is a pigment that gives hair its color, and the redheads have pheomelanin instead of melanin. Now we will talk about the cuticle, which is the major component of a hair structure. The cuticle varies in the number of scales, and under scales we study the how many number of scales are present in per centimeter, how many scales overlap, the overall shape of the scales, and how many scales they protrude from the surface. The cuticle also varies in the thickness, and it can also be differentiated on the presence and absence of the pigments. The characteristics of the cuticle may be important in distinguishing between hairs of different species, but these are often not useful in distinguishing between different people. Simultaneously, the cortex part of the hair, it also varies in the thickness, texture, and the color of the cortex. The distribution of the cortex is perhaps the most important component in determining from which individual a human hair may have come. The microscopic examination can also reveal the condition and shape of the root and tip of the hair. The next part of the hair structure is medulla. The medulla also varies in thickness, continuity, and opacity. During the examination of the medulla, one should see that the medulla is a continuous structure or it is broken into pieces. Opacity is the characteristic of medulla which reflects that how much light is able to pass through it. The medulla, it may also be absent in some species. Like the cuticle, the medulla can be important for distinguishing between hairs of different species, but often does not lend much important information to the differentiation between hair from different people. Here you can see a diagram in which the continuity of the medulla is shown. In the upper example, the medulla or the structure of the medulla is discontinuous or it is broken into pieces, where in the lower diagram, the medulla is a continuous structure. This diagram is showing you the microscopic view of hair structure. In the hair structure, the cortex, cuticle, and the medulla layer is shown. The cortex is the middle layer which consists of the longitudinally arranged elongated cells, whereas the medulla is the inner layer and composed of the keratinized remains of the cells, and the cuticle is the outer layer and consists of the thin non-pigmented scales. In addition to the three parts, the hair also consists of different granules of pigments, ovoid bodies, and cortical foci. Now, I will talk about the formation of the hair and its growth. Hair formation begins in the third month of the fetal life. Each hair grows through a follicle and it is made up of epidermal cells that grow under the dermis. Initially, the epidermis thickens and cells begin to grow down into the dermis. This downgrowth forms a cap over some of the connective tissue to create papillae, whose cells multiply to form the hair. As these cells are pushed up the central canal of the hair shaft and away from their source of nourishment, they become impregnant with the keratin. The morphogenesis of most of the hairs follow a cyclic pattern of cell proliferation and cell differentiation initiated in the mid to late embryonic development, and this process is repeated throughout the life. Now I will tell you about the growth of hair. There are basically three stages of hair growth. which is determined by the root of hair. The root of hair is the proximal end of a hair strand. There are three stages. The first stage is the anagen root stage. In the anagen root stage, this stage is an active growth stage and often found with a ribble-like tip and a follicular tag, that is a soft tissue from the follicle. The next stage that comes after the anagen root stage is the catagen root stage. The catagen root stage, it is considered the intermediate stage of hair growth, and after the catagen root formation, the next stage comes telogen root stage. The telogen root stage is a resting stage and has a bulb-like shape. This stage often found with very little pigment and abundance of cortical foci. Now, I will talk about the different types of hair. Humans have several different types of hair that can be classified depending on their body position and hair form. Generally, three different hair types are found. They are based on the length and diameter of the hair follicles in the skin, and these are called lanugo, vellus, and terminal. The lanugo is the hair type produced in the very first cycle of the hair growth when a hair follicle enters shortly after it develops in the embryo. These hair are fine and soft, and they grow all over the baby's body, and its main function is to retain body heat. They all grow at the same rate and so are of the same length. Around the 8th month of development, this hair is usually shed, and often a second generation of lanugo hair then starts growing and lasts until the first 3 or 4 months of extrauterine life. These hairs are then replaced by the vellus hairs. The vellus hair is short, fine, called as peach fuzz unpigmented hair. The vellus hair is very soft, much softer than the lanugo, and grows in most places on the human body in both sexes. It is usually less than 2 centimeters long, and the follicles are not connected to the sebaceous gland. The vellus hair is also present among pre-adolescents. The third type of hair is terminal hair. The terminal hair is developed hair which is generally longer, coarser, thicker, and darker than the vellus hairs. The phases of growth in terminal hair are most apparent than in vellus hair, and the former generally has a longer anagen phase. The terminal hair contains a large hair follicle and sometimes a medulla. During puberty, many hair follicles in the pubic region, armpits, legs, chest, and face, the latter two in case of males, transform from vellus hair to terminal hair under the influence of hormones. The terminal hairs can be further subdivided into different types depending on their nature and the position of growth on the body. The different types of terminal hairs include hair of the eyebrows, eyelashes, head hair, beard hair, and pubic hair. The forensic anthropologist compares the various morphological characteristics of the hair samples such as medulation, diameter, form, and cross-section. The identification of body area can also be done from the hairs. The body area from which a hair originated can be determined by the general morphology: length, shape, size, color, stiffness, medullary appearance, curliness, and microscopic appearance all contribute to the determination of the body area. There is a wide range of interpersonal variation in head and pubic hairs, as a result of which the majority of work in forensic anthropology has been in comparing and differentiating hairs from the head and pubic regions. The head hairs, they can appear uncut with tapered tips, but they are most often cut with scissors and razors. In general, these hairs are subject to more alteration than hairs from the other body area. Alteration can be attributed to the use of chemicals such as hair dyes, rinses, permanents, and frost, and they are also attributed to the use of or different kind of environmental conditions such as exposure to the excessive sunlight, wind, and dryness. The known sample usually contain a random sampling of hair from different areas of the scalp, whereas the pubic hair, they commonly possess tapered tip, but they also be cut with scissors and razors. The buckling is a special characteristic found in the pubic hair. The pubic hairs are typically coarse in diameter with wide variations. Now we'll talk about the limb hairs. The limb hairs include hairs of the arm or leg hairs. The limb hairs, they are fine in diameter with little variations. Their structure is like an arc and appearance is gross. The limb hairs tip are usually tapered, often blunt and abraded, and they have rounded scales which end due to the wear and soft texture. Here in this example, you can see the beard hair where the diameter is very coarse and they are very broad and the texture is stiff. Whereas the facial hairs, which include hairs of beard and mustache, they are very coarse in diameter with irregular or triangular cross sections. The facial hairs, they have very broad and continuous medulla. Their medulla may be double and the facial hairs texture is stiff. The chest hairs, they are moderate and variable with shaft diameter. Their tips often darker in color. The chest hairs are long and fine. They have an arc-like structure with gross appearance. These hairs, they may have granular medulla and their structure is stiff. Next is the axillary or the underarm hairs. The axillary hairs, they resemble pubic hairs in general appearance, but these hairs are comparatively less buckling than the pubic hair. The underarm hairs, they have the similar medulla and they have fine tip. The other body hairs, which include eyebrows, eyelashes and trunk hair. The eyebrow hairs, they are stubby and they have saber-like appearance, whereas the eyelashes, they also have stubby and saber-like appearance and they are short. And the trunk hairs, they are the combination of limb and pubic hair and also referred as transitional hair. The forensic anthropologists routinely try to establish the racial affinities of the head hair samples. The main features of the hair that are studied are the shape of the longitudinal cross-section, hair pigmentation, shape of the cuticle, hair texture, medulation, and hair form. In case of Caucasoid, the cross-section of the hair varies from the round to oval with an evenly distributed pigment granules, a color ranging from blonde to brown to black. They have the medium cuticle, fine to medium coarseness, discontinuous medulla, wavy hair form, and their pigment density, they vary from light to the moderate, and they have moderate shaft diameter with very little variation. Now I will discuss about the hairs of Mongoloid races. The hairs of Mongoloids show a round cross section and a straight hair form. They have a characteristic reddish appearance or tinge. Their hairs are often coarse, having a wide diameter than those of the other racial groups. The cuticle is usually thicker than that of the Negroid and Caucasoid hairs, and the medulla is continuous. The hair shaft or the cortex of the Mongoloid hair contain pigment granules that are generally larger in size than the pigment granules of Caucasoid hairs. As you can see in this diagram, the Mongoloid hairs, they are coarse, they having sticky pigments with heavy density, and their cross section is round. The Negroids, they exhibit a flat cross section of hairs with dense pigmentation, a thin cuticle, absence of medulla, and a curly, woolly, and kinky hair form. The Negroid hair pigment are larger than those found in other racial groups, and their shaft exhibit variation or apparent variation in diameter, as it is often flatter nature and the manner in which it lies on the microscope slide. The twisting of the hair shaft or the buckling may be present, and the hair shaft may be frequently split along its length. This diagram is showing you the example of Negroid hair where there is prominent twist and curls, and they have heavy pigmentation, and the cross section is flat. The determination of sex can also be done with the help of human hair. Sexing of a human hair is although difficult, except that of beard and mustache hair. Male hair is usually thicker, coarser, and darker. In human hair, bar bodies are found in hair follicles in a proportion of 29 plus 5% in the female and 6 plus 2% in the males. Now I will tell you about the age determination through hair. Age can be determined sometimes from the hair, but only within wide limits, as between that of an infant or an adult. Root of hair from children will dissolve rapidly in a solution of caustic potash, but In older people, roots will resist this treatment. Age can be determined through hair diameter. If the hair diameter is 0.024 millimeter, then the age of the person would be 12 days. If the diameter is 0.036 millimeter, then age would be 6 months, and 15 years old would have a diameter of 0.053 millimeter, and adults with 0.07 millimeter. The body hair of the human fetus and newly born child is fine, soft, non-pigmented and non-medulated. The lanugo hair is replaced by the hair which is coarser, pigmented and medulated, and it has a complex scale pattern. At puberty, axillary and the pubic hair grows, which is at first they are fine, curly and later becomes coarse, pigmented and spongy. Adult hairs have maximum pigmentation. Adult hairs have maximum pigmentation, and the loss of scalp hair in men starts from the third decade. In women, there is often loss of axillary hairs and increase of facial hair at about the menopause. Grey hair usually appears after 30 years. Now, I'm going to tell you about the determination of hair treatment. Morphological examination can identify whether a hair sample had undergone artificial treatment such as use of dyes. Moreover, as the growth rate of head hairs is known, the investigator can assess the approximate time of this treatment by measuring the length of the untreated area of the hair. This is done by a direct side-by-side comparison of the color of the questioned and known artificially treated hair. Students, now we will study the forceful removal of hair. A microscopic study of the hair root determines whether the hair was forcibly removed from the body or shed naturally. Hairs that fall out naturally exhibit a club-shaped root, while a forcibly removed hair will be stretched and may have tissue attached to it. The manner in which a hair was removed has considerable forensic value, especially when there is a possibility of violent contact between a suspect and a victim. Microscopic examination can also be used to establish whether the hair was burned, cut or crushed. Sometimes forcibly removed hair with tissue tags or roots are found at a scene. The tissue tag is a portion of the hair follicle from which the hair grows. The tag is the most time visible to the naked eyes or else under a microscopy. If the tag is present, then the serologist may add several more tests to the microscopic hair examination protocol. The blood groups of a person can also be determined through hair. The ABO groups can be determined in a single hair from any part of the body by a modified absorption elution technique with 100% accuracy. Now I will talk about the determination of hair identical with the hair of victim or the hair of suspect. By careful comparison, the examiner can say that hair could have come from a particular person. Debris, grease, they are adherent to the hair, and it is very important in the examination of hair. Microscopically, the intimate structure shows uniformity in general shade, which is not seen in hair of natural color. Because of diet and drug intake and atmospheric conditions, traces of 18 elements are deposited in our hair in proportions quite unlike those in other persons, which can be measured through neutron activation analysis. Now we will talk about the determination of cause of injury. Hair can be used to determine the cause of injury. If the hair has not been cut, the tip is pointed and non-modulated, but repeated injury to the tip damages the cuticle, due to which the exposed and unprotected cortex splits and frays. The hair of axilla and pubis has frayed ends, so they can also frequently brushed hairs. Blunt injury and crushing with heavy blunt scissors crushes the shaft with flattening and splitting. A sharp weapon produces a clean, uniform cut surface. After a week, the end becomes square, smooth, and later rounded, but it becomes blunt. After 3 to 4 months, the end becomes elongated, but the medulla is absent. Hair may get scratched or sined due to burns or firearm injuries. Sined hair is swollen, black, fragile, twisted, or curled, and it also has peculiar odor. Carbon may be found deposited on this kind of hairs. The tip is also swollen like a bulb. Now I will tell you about the medico-legal importance of the hair. Hair is important in crime investigation. It remains identifiable on both the body and the alleged weapons in crimes committed long before. It often provides the only connections between a weapon or even accused and the victim of an assault. Motor vehicle responsible for injuries may be identified by the detection of hair on the vehicle. In the case of rape, the pubic hair of the accused may be found on the victim or vice versa. Stains on the hair may indicate the nature of the assault, for example, mud stains in struggle, seminal stains in sexual offense, salivary stains in injury. Stains may be available from the walls, doors, furniture, and may indicate the scene of crime. Hair is important in identification, especially where there has been some known peculiarity of the hair, dyeing, bleaching, or artificial weaving. In chronic poisoning with heavy metals, for example, arsenic, the poison can be detected in the hair. The time of the death can sometimes be determined from the growth of the hair on the face. The shining of the hair indicates burn or close range firearm injury.
The hair is also helpful in differentiating scalds from the burns. Age of a person may be determined from the growth of the hair on different parts of the body. Sex may also be determined from their distribution on the body and texture, and the nature of the weapon can be made out from the injuries to the hair.
So students, let us summarize what we have learnt from this module. Human hair has a significant potential in forensic anthropology, and it has been extensively used in forensic investigations. The type of hairs recovered, the condition and number of hairs found in the scene of crime all have great relevance as evidences in criminal investigations. The forensic anthropologist routinely compares the morphological characteristics of the hair samples in question to the known hair samples to determine a transfer. Human hair has also been successfully used to assess drug and substance abuse, as drugs of abuse cannot be often detected in body fluids. Using very sophisticated analytical techniques such as immunoassay and gas chromatography, investigators are now extensively using human hair to solve cases of poisoning and drug and substance abuse. The forensic anthropologist, he compares the various morphological characteristics of the hair samples such as medulation, diameter, form and cross section to determine age, sex, ABO blood groups, body area and race. The examination of hair is also one of the important parameter for medico-legal point, and this medico-legal point of view, it can be used in identification, sexual offences and crime where the hair or hair fibers are found, or it can also be used in hit and run accidents or chronic poisoning by metals. Thank you.